The overall goal of this procedure is to use human cord blood samples to isolate pure CD34 positive cells to study the effects of specific drugs of interest on megakaryocyte differentiation, pro-platelet formation, and platelet production.
This method can help answer key questions on the megakaryocyte and platelet field, such as what are the effects of specific treatments on megakaryocyte differentiation and platelet production and function. The main advantage of this technique is that it provides a method for the consistent isolation of viable CD34 positive cells from fresh umbilical cord blood.
Though this method can provide insight into megakaryopoiesis, other cell types such as lymphocytes, erythrocytes, and macrophages can also be studied.
Begin by adding 10 ml of separation buffer into one 50 ml conical tube per 10 ml of cord blood. Using an 18-gauge blunt needle mounted on a 10 ml syringe, transfer 10 ml of blood into each tube, followed by the addition of 15 ml of lymphocyte separation medium to the bottom of each tube. Separate the cells by density gradient centrifugation, then at the end of the spin, carefully collect 5 to 10 ml of mononuclear cell-containing buffy coat layer into one new tube per sample. Bring the total volume in each tube up to 50 ml with fresh separation buffer, and centrifuge the cells again.